University of Oregon and Thermo Fisher Scientific. I've met many of you at this point, but I'm Darren Johnson, Director of the Material Science Institute and Professor in the Department of Chemistry and Biochemistry here at U of O. It's been a lot of fun organizing this with our counterparts. We got David Piper on the line. I don't know if Alexia's here also as one of the co-organizers. I've got the Zoom screen. There's too many people now for me to see everybody up there. So welcome and thanks to our speakers this week. As I mentioned, people were coming into the meeting, well, since we're on Zoom this week instead of in person, like we were last time, we'll save questions maybe to the end. You can either ask by posting in the chat and we'll get those read to the speakers if they can't see them with screen share, or you can raise your hand and we'll call on you. And so with that, you can see that Ghee Ong will be our first speaker. You should be seeing his cover slide. Ghee is a professor in the Knight campus for accelerating scientific impact here at U of O. He is an electrical engineer by training. He got his bachelor's, master's, and PhD at the University of Kentucky, and started at his academic career at Michigan Technological University in 2006. In 2019, the Knight campus was able to recruit him, and he's one of the first faculty members to start up his lab in the brand new building. You can see it in there's a little bit of the image of the building behind him. If you've been to the campus, you might recognize where he is. I think probably overlooking the terrace. And I guess now 3 years, Ghee, that you've been here, time sure flies fast. You set up a number of collaborations across campus and nationwide, and continued ones you already had going nationwide, including a new one with my group. We've done some work with you, and where you're really helping us on sensor device fabrication and testing. Ghee's also been instrumental in setting up a lot of the curriculum on the Knight campus. As you can imagine, there's a heavy lift in setting up a new biomedical engineering program at a university that's never had an engineering program before. So many thanks to Ghee for all of his efforts in that over the few years he's been here. Ghee's area of expertise includes implantable sensors for regenerative medicine and sensor technologies for cell manufacturing. And today he's going to talk to us about wireless implants for, I can read on the screen, not on the sheet of paper, wireless implants for in situ biofeedback after tendon and ligament repairs. So Ghee, look forward to your talk.
Well, thank you. Can you guys hear me, right?
Sounds great.
Okay, good. Well, thank you, everyone. Good afternoon. And then, first, thank Tony and Darren for inviting me. I'm very, very happy to be able to describe my latest research finding to this group. And then, again, my name is Keat Ghee Ong, and I'm the professor at the Knight campus. So before I start, I need to disclose a potential conflict of interest. I am the owner and also the founder of Pandera Technologies Incorporated. The company was formed in 2020 as a way to commercialize my lab research. As you know, the Pandera Technologies and the university, the work that I present today are actually overlapping between these two, as the university research team are developing a lot of early stage proof of concept technologies, and the company is actually moving on to working on testing and also moving the technology more to the commercial space. So this is my conflict of interest disclosure statement. Okay, I know that Darren just described these slides and I prepare for it. Again, I'm consider myself as a hardcore engineer. And after I get my PhD in 2000, I actually worked in a couple of small startup company working on a different medical devices, develop new medical device technology for different applications. And then 2006, I joined the faculty rank at Michigan Technology University before I joined UO at 2019. Now, for those of you who do not know where Michigan Tech University is right in the middle of Lake Superior, and it has a lot of snow. So this is a picture I like to show people. Let me see if I can. Yeah, this is a picture I like to show people. And this typically happens between December to March. And I keep telling people I'm quite happy to move to in Willamette Valley, which we don't see snow as much. So I don't have to wake up 5 a.m. in the morning, spend an hour and a half to shovel snow from my driveway. That's a good move, I think. So let's talk about my research area. For the last 20 years or so, I work on different type of technology, different type of research, most of them focusing on sensor technologies and medical applications. Now, if I want to categorize or group my research together, I can say that my research are primarily three major groups. One of those is the one that I mostly focus on today is medical implantables. And I also have a lot of background in medical diagnostic devices, especially point of care diagnostic devices, and also biosensors, and of course, wireless sensor technology. Now, today, my talk obviously focusing on medical implantable devices. So why my lab focusing on implantable devices? Well, the reasons can start with looking at the implantable systems that we have on the market right now. So let's look at the ones on the left. We are seeing insulin pumps and heart pacemaker. And what are these two have in common? Well, in today's world, these type of advanced implantables have a lot of capability. Now, one of the capability I really like, and I think it's very important, are their self-monitoring capability. So not only they can measure a lot of physical or biological condition of the patients in real time, they can also monitor the condition of themselves and how they operate and collect a plethora of data for the physicians to gather to understand. Of course, they also have fancy things like data storage and wireless connectivity. But the more important thing is they have ability to provide data for the researcher to study and for the physician to improve the treatment and so on. Now, let's look at the counterpart of these advanced medical implants. We are talking about things like bone fixation device and screws. Now, these type of spectrums of implants are very simple. They are nothing more than just a piece of metal or plastic providing mechanical support. Contrasting to the one that I showed before, they have no self-monitoring capability. What it means here is once you put this into the body, and that's it. It's very difficult to see what happened. You can go through some imaging technique, MRI, X-ray, but you are not going to be able to measure the condition like stress and strain and mechanical pressure on it. There's nothing no easy way to get that. It also means that there's no way to detect if they are performing properly, and there's also very little way to getting the data to further improvement the treatment or further understanding the disease. So I do believe that we can do better with these implantable systems. We can put sensor on them to make them multifunctional. As I said before, we really want to focus on the simple implantable devices, simple things like bone plate and bone screw, and there are a lot of sensor technology that can do for that. We can talk about, we can use things like complex sensor that has Bluetooth systems or have different type of wireless transmission scheme, but they are not practical for this type of simple implants. Because, for example, if you look at that, the simple implants itself doesn't have internal batteries. So it doesn't make sense to have a sensor that requires internal batteries incorporated to that and complicating the whole implants. So that's why that we focus a lot on battery-free wireless sensors. Now, for this type of battery-free wireless sensor that we work on, the sensor doesn't have internal battery, as I described. It actually receives the power wirelessly through electromagnetic coupling. For this type of sensor, typically they will stay dormant in the body if they do not receive electromagnetic power. And if you want to know what this technology will look like, well, if you can just pull out your today's credit card and you look at the tiny chip, the RFID chip, it's similar to that, the concept-wise. Of course, it's completely different design and different type of structure, but the way is the same. You scan it, and then it will tell you what information that you need to know. This type of technology is also smaller, safer, and as I said before, very ideal for simple implants like orthopedic implants. So why do I want to focusing on orthopedics? Well, especially right now, I'm focusing on soft tissue in orthopedics. Well, there is a big market need, and I think that today orthopedic monitoring is very, very underserved. There are not a lot of technology that can do that. Just look at the market size and the need. Tendon and ligament injuries are very, very common. And just a couple common injuries we know. For knee, you have ACL tears, shoulder, rotator cuff injuries, and ankle, you have high ankle joint injury or syndesmosis injuries. And the number is actually not small. You look at rotator cuff surgeries, 300 per year, and then the healthcare cost is 3 billion. And not only that there are a lot of them occurring, but another issue here is a lot of the repairs or surgeries are not very successful. For rotator cuff surgery, 25% of them fail. For patients 65 or older, 77% of them fail. So it means that more than half of the surgery fail. So obviously there's a need for better monitoring or better approach to improve this type of surgery. Similar with ACL, you have 200,000 injuries and the cost is 40,000 per year. And this number was collected in 2014. In today's high inflation world, actually I do not know the number, but I'm pretty sure it's much higher. And this is the picture of Tiger Woods that someone sent to me, and I like to stick into my slides. It just shows that how much orthopedic injuries that can happen to anyone. I mean, this is Tiger Woods, a very active person, and he has all types of injury. You have joint injury for neck, knee, legs, spine, and you can imagine this is basically the indication or the marker that we will need to resolve or address. So what can orthopedic sensor do? I mean, okay, so we can measure conditions at the injury site, but what can we use for? So what? Well, one thing that I know that it will be useful is for rehabilitation, improving the rehabilitations. And right now, today, there are very few ways to actually gather information to provide biofeedback during rehabilitation. Today, for physical therapists, if they want to gauge, what type of physical therapy treatment to give the patient, they use a conservative approach. They give the timeline, means that you have to wait 6 weeks for that, and then ask the patient, are you still feeling pain? Can you lift your arm to this level? If it's okay, then go ahead. So doing this sort of conservative approach, of course, there are always some issues. Well, first of all, it takes a long time. Sometimes it's longer than needed. And you also need more physical therapy sessions, means it's going to be more expensive. And a lot of times for orthopedic healing, time is an essence, means that you should not wait too long, and you should not be getting too early to exercise the injured site. And if you're waiting too long, you actually not get the best outcome. So it is always useful to know the exact time point to start the physical therapy. And there's also a situation where some patients that take too long to heal, and even the conservative approach is not conservative enough, that if you start the physical therapy certain session too early, you can actually re-injure the injury, and you can make things worse. So real-time biofeedback is going to be very, very useful to tell the physicians when to start the certain type of physical therapies. And that's what our lab is focusing on, and that's what I'm going to talk about today. So let me change gear a bit and describe the technology. And there's a lot of technology can be used for, but today we're focusing on, and let's see my lab, we're focusing on a wireless passive sensor, means that it doesn't require battery, that is based upon inductor capacitor resonance circuit. For this type of sensor, it has a couple components. It has an inductor here, capacitor, and resistor. So for those of you who have some background in electrical engineering, if you combine these three together, it will form a circuit we call resonance circuits. Now, when they are forming a resonance circuit, and if you have a coil here, it's basically just a loop of conductive copper wire. You're passing the current through this loop, it will generate electromagnetic field, and the electromagnetic field is going to interact with the sensor. And the sensor is going to generate back another electromagnetic field, and this magnetic field, electromagnetic field, is going to induce another voltage at the end of the coil here. So we can just measure the voltage at the coil. From there, we can back calculate the resonance of the sensor. And this is how the wireless communication between the sensor and the detection coil happen. This is a typical resonance curve. And as you can see here, we generate a frequency varying signal, and then the sensor generate back the frequency varying signal. And this is what we measured at the end of the coil. As you can see here, there are nice resonant peak right here. And a couple important information or parameter we can use. And the middle of this is what we call the resonant frequency. And this is basically the highest point, the position or the frequency that correspond to the highest point. And another point that another parameter we like to use also, we call it the quality factor, is basically how sharp this resonance is. And in this case, the gap between these two points is can be used to calculate the quality factor. If you have a resonance that's very sharp like this, we call it has a very good quality factor, means that the quality factor has a very high value. If you have another curve like that, very broad resonance, then the peak is like this and this, then this is going to have a very low quality factor. And we can also track this to indicate the change in some of these in value. So the sensor can be used for measure a lot of different type of situations in terms of orthopedic injury. And one of the applications we use for is to monitoring suture tension at rotator cuff repair. Now, for rotator cuff injury, when the surgeon needs to do the surgery, normally what they do is they're using the suture to connect the rotator cuff. As you can see here, they connect at this point, they use the suture to loop through this rotator cuff, and there is a tear in the rotator cuff right here. So they use a suture to pull this and then anchor it to the bone. So with the suture, it means that all the tensions, all the load that apply to the original rotator cuff is being shared or is being supported by the suture right now. So that the rotator cuff can have a chance to heal. And as you can imagine, as the rotator cuff start to heal, means that the rotator cuff is going to take back some of the load sharing. It's going to get more and more load as this one is getting better and better. So if I'm a physical therapist, what I need to do is I just need to look at tension at the suture right here. And if the rotator cuff is healing properly, you are expecting the tension at the suture is going to decrease over time, indicating the rotator cuff is healing better and better. And of course, if it doesn't decrease, it means that the rotator cuff tear is still significant, it still hasn't healed yet. We can use the information to gauge when should we do strengthening rehabilitation exercise. And on the other hand, if the patient has very bad rotator cuff, there's a chance that the suture here is going to tear through the rotator cuff. And when that happens, you can imagine that when this is tearing through the rotator cuff, the tension at the suture is going to decrease immediately. So the same thing, the surgeon can look at the tension at the suture, and if they see a sudden decrease, it means that the surgery or the suture has failed. So using this type of strain monitoring or the tension of the melting of the suture tension, we can also use it to detect potential failure of the suture. So how do we measure the suture tension? Well, we are actually not measuring at the suture, but we are measuring at the ankle side. As I've shown you before, the suture is being looped through the suture anchor. So it means that all the tension applied to the suture will transfer to the anchor. So what we need to do is we just need to instrument the anchor, putting the sensor inside the anchor. So as the suture is pulling the anchor, we know the force from the suture, and then we can back calculate what the suture tension is. And this is our design here. We have the same component, inductor, capacitor, and resistor. The inductor is the coil right here. The capacitor is a tiny thing right here. And the inductor is, the capacitor here, and the resistor is a strain gauge they're connecting right here. And this is the blowout of the design. This is the inductor. The capacitor is somewhere glued right here. And then the strain gauge is right at this spring, this S-shaped spring. In practice, the surgeon is going to loop the suture through this hole. And then the suture is pulling on this S spring as the tension is being applied. Now you can imagine that what happens when the tension is increasing, right? As the tension is increasing, you actually see the S spring being extended, and you actually create a strain on the strain gauge. When that happens, the resistance of the strain gauge, which is the R here, is changing. It's going to change the resonance profile of the sensor, and we can capture that wirelessly. And this is one of the results. You can see there are 3 different sensor designs. These are just 3 sensors with a different type of inductor and then different type of spring constant on the spring. And we just want to showcase that here we can have a different resonant frequency. So we can choose the resonant frequency we want to operate. We can allow multiple sensor putting in the near proximity, and we can still know what is the tension at each suture, so we don't confuse with them. And by using the tension, the spring constant of different spring constant, we can actually have the suture anchor with different sensitivity. For example, for this one can be used for something that has that require very high sensitivity but less dynamic range. So we can develop different type of suture anchor for different needs. Not all suture, not all orthopedic surgery use a suture anchor. For a lot of orthopedic surgeries, they use something called suture button. A suture button is basically operate just like the button in our shirt. What it does here is it create the suture loop through the button, and then you can pull on the button, and the button typically rests on the opening. In this case, the opening on the hole. A hole on the bone. So, for example, we can the suture button is being used for ACL repair and also syndesmosis repair. And this is the typical applications or this is typical ACL repair. You have a tendon here, and then you have a suture pulling through the tendon, and the button is at the end. And the suture is actually looped through the button, and the button is rest across the hole drilled by the surgeon. And as this joint is being moved, is actually pulling on the tension on the suture, and the suture is tense on the button. The same thing with syndesmosis repair here as here. The suture button is using as a securing point that the suture is going to be providing the force that can tense on. So this is the design of our suture button sensor, and it contains of 2 components. The traditional, the standard suture button, which is right here, is the 2 hole, and then you have a suture loop through this. And another component is the sensor here. The sensor is basically a tray that the suture button rests on. And this is the blowout of the sensor. It has the same thing, inductor, capacitor, and the strain gauge. And it also has something we call the load plate. The suture button is actually going to rest on top of the load plate right here. And there are 2 tiny ridges at the load plate in the middle of it. As you can imagine, as the suture button is resting on the load plate, and if you are pulling the suture, it actually create a tensile force at here. These ridges is actually going to bend this load plate like this way. As it bends the load plate, what it happens is it's actually going to tense the strain gauge. It actually causes a bending motion at strain gauge. When that happens, you actually change the strain gauge value, and similarly, it's changed the quality factor of the resonance, like what we shown before. And again, that's what we have. We have a change in the quality factor as we apply the load across the suture button. And we also do a lot of repeatability tests, show that this type of design is very stable across multiple loading cycles. So what I showed you before are 2 types of implementation of our sensor to measure suture tension post-operative. However, those applications require bones, means that we need implants that can secure into bone or around bone. Not orthopedic surgeries rely on bones, they involve bones. There are certain type of injury, for example, Achilles tendons, that the fixation is nothing more than just a suture fixing or connecting these 2 torn tendons. It doesn't have bones, it doesn't require suture anchor, it doesn't require suture buttons. So what it means is our old design, they require strain gauge, capacitor, copper wire, is not going to be applicable here because they are not biodegradable and they are not biocompatible. So we need a new type of design. The new sensor needs to be biodegradable and has to make with biocompatible materials. And there is our new sensor comes from. We are actually building our sensor using what they call a surgery device called suture pledget. Sometimes they are called surgical pledget. Surgical pleasure is nothing more than just a piece of material, a patch that is used to loop through the suture so that it can create more surface area so that when you pull the suture doesn't tear through the tissue. It's basically just a securing like a washer or the screw head that you screw on the wall. So what we are designing here is we are instrumenting our sensor on the suture pleasure. And as suture loop through the pleasure, so as you tense the suture, it will transfer the force or the tension on the pleasure, and we can sense that, we can back calculate and understand and determine the tension at the suture. And this is our new design. Again, we are not using capacitor, we are not using a string gauge anymore. What we have right now is nothing more than just a loop of conducting pattern in this spiral shape. This is actually a spiral inductor. Now, when we design it properly, this spiral inductor will actually resonate just like the RLC circuit I shown. What it means here is, if you put a coil nearby, you're going to generate a resonance profile just like what I showed you before. That's because the inductor itself has some parasitic capacitance, and then the conductor has some type of residual resistance. It can behave like RLC circuit. And this is the design that we use. We develop the pleasure. So we have a pleasure in the form of this, and the suture is looped through this hole. And the bottom part of the pleasure is a solid piece, but the inductor pattern is embedded in the top surface or the top portion of the suture pleasure. So as we are pulling the suture and we tense it, what happened to the pleasure is we are actually bending the top surface here. And when the top surface is bent, it actually bends the inductor. And for inductor, if you are bending the inductor, it means that you are changing the dimension of inductor. And when that happens, you're actually changing the resonant frequency of its resonance. And this is exactly what happened. We are building the suture pleasure. We are connecting in the mechanical tester, and we are applying loading on the suture. As we apply the load, we actually bend the copper inductor patterns, and then it changes the resonant frequency, and is quite repeatable, as is shown in this figure here. Okay. So as shown, I just showed you a couple design on implantable sensors used to measure suture tension after the surgery. Now, the reason we focus on that is because there are a lot of interest right now to improve orthopedic surgeries using therapies that are based on biofeedback. And right now, a lot of people are focusing on developing better approaches using AI, drugs, and materials to make the therapy better. However, in my opinion, we still need biofeedback with good sensors. And that's what my lab is focusing on. We are focusing on building simple sensors that can provide biofeedback, and the sensor that we develop do not require secondary surgery to remove it, is safer, and is less expensive. And then with that, I'd like to thank my collaborator, and without my lab members, this project would not happen. And also, I want to acknowledge the funding that helped to develop all these technologies. With that, I guess we'll wait until the second speaker before we can ask more questions.
Thanks, Guy. Great talk. And actually, if it's okay with you, we could allow for some questions now while it's fresh in folks' minds.
Yeah.
Be okay? Okay. So if anyone has a question, please raise your hand or post it in the chat, or just say in the chat you've got a question. We'll call on you.
So how different would the surgery? Sorry, how different would the surgery be with one of these devices, where as opposed to a more traditional, surgical approach? Is it significantly more difficult? And is it significantly more costly, or is it more risky?
Yeah. So that's very good question. Yeah. Yes. We work with a lot of clinicians on this project, And our main goal is, well, that's what they told me. They say anytime when you want to change a surgical procedure, it's a no-go. They don't want that. They don't want to change anything at all. So that's what our focus. That's why that the suture buttons and the suture anchor I showed you, they are based upon the same design or very similar design as today's suture anchor and suture button. Okay, the dimension, the way they implement it is the same. Except maybe we are slightly larger or slightly longer, it's a slight change in dimension. So, and the only difference is after you do the surgery, when you want to detect it, you have to wear a wearable device outside. So in terms of a surgeon, there's no change in procedure. Now, in terms of safety, we try not to use battery so that because we want to improve the safety. And we still need to go through, and that's what the Pandera technology is coming for. They are going to do a lot of mechanical tests, make sure that it doesn't break. As long as we can maintain the mechanical integrity of the suture anchor, suture button, then the safety will be preserved. And that's our regulatory approval strategy. That's how we're going to go for that.
Is there any need to remove it?
No,
later?
That's why we don't use battery, because they do not want to remove it. And a lot of surgery is permanent implant. They cannot remove.
Right.
So there's a question. Talk about, are they compatible with MRI? Yes, we are not using any magnetic material. So a lot of the material we use is similar to the suture anchor or suture button they use. So there are certain, they're still going to have a certain, we call that, what's the word I want to use? The MRI effect, they become brightened because they are metal, but it's not going to be more than using the traditional suture anchor.
And in all the cases that you showed, was the strain that the suture is applying, in all cases, was it changing the inductor? Is that how the resonance was changing in all?
Yeah, there are different design. For the suture anchor and the suture button, the change is actually the resistance, because you are seeing a strain gauge. So the inductor and capacitor stay constant. But the other one, the suture pledget one, we don't change a lot of inductor. Well, we don't change a lot of inductance, but we don't change a lot of resistance, but we change the inductance. That's why we are tracking different things, right? For the suture anchor and suture button, we are tracking the quality factor, because the resistance change the quality factor, not much on the resonance. But for the pledget, we track the resonance frequency.
Got it.
I see one other question in the chat. Are you able to see the older chat posts?
Oh, okay.
I can read it to you also.
Oh, okay, yeah, I got it, yeah. So
Okay.
the question here is that, do you expect the post-op readings require a visit to PT, or could there be a clothing app design allowing patients to record long-term data? That is a very good question. And our strategy right now is earlier phase, of course, the patient has to go to a PT to understand the data. But we actually, right now, on my Samsung phone, we actually have the app, and we actually have a device that we can connect to that can capture the data onto here. So we can measure the response, we can measure the resonance frequency. The only thing we're missing right now is what does the frequency mean and what does the strain mean for our rehabilitation. That is a data that we still rely on our clinical collaborator to collect to make the correlations. So yes, the technology can be cheap enough that you can use it for in-home monitoring. And if you are an athlete that you are really crazy about how well your body performs, you can actually track it as you run. Just like wearing an Apple Watch, but you're wearing a special watch that can track your suture.
What was the frequency or duty cycle for response? So how often does it send a signal?
Yes, that depends on the device itself. If your device can generate a very high and fast frequency, we can detect, I don't want to go too high, but between 30 to 60 hertz is what I expect to be maximum. Anything higher than that is probably do not provide a lot of useful information anyway.
Yep. Just thinking about like that impact.
Yeah.
Is it going to be rounded off because it doesn't respond quickly enough? Okay.
Yeah, we should be able to capture some of the impact. because right now we are tracking the whole resident profile, right? But once you know the resident profile, you know their range, you can actually do a point measurement, and that can be very fast. You just focus on one point. As you shift left and right, you can use algorithm to estimate how much you shift.
Okay.
And then with that, you can do very fast. And the system can do, up to, a kilohertz or so.
Sounds good.
Great, thanks, Guy. I don't see any other questions, so thank you for an excellent talk. If anyone thinks of any other questions for Guy, I'm sure you can reach out privately in the chat too while we have some time still today. With that, we will shift gears to our second speaker. Ryan, feel free to share your screen if you want, as I'm doing a brief introduction. It's my pleasure to introduce Ryan Bomgarden from Thermo Fisher Scientific. I met Ryan about 40 minutes ago from across country, so great to see you at our session. Ryan's leading a team of oh, sorry, what's that?
No, I was just saying thanks for having me.
Oh, yeah, of course. Ryan leads a team of scientists in a mass spec reagent group focused on developing novel products and workflows for the proteomics market. He got his bachelor's degree from Coe College and in both chemistry and molecular biology, and then obtained his PhD at Stanford University, characterizing checkpoint kinase binding and signaling. Ryan joined Pierce Biotechnology in 2005. So I learned as we were getting together earlier today that places you in the Rockford, Illinois site, which I haven't had the pleasure of coming out to visit yet, but thank you for joining us from afar. A nice thing that Zoom is enabling. And is now part of the Thermo Fisher Scientific company. So Ryan, thank you very much for joining us. Now, I guess in the early evening, your time probably, and we're looking forward to your talk on providing COVID-19 clarity using TMT reagents for precision measurements.
Yeah, no, thank you so much. I hope you guys can see my slides, and I think we're ready to go. Yeah, so again, going to be quite a different talk from the previous speaker. I mean, we're going from mechanical to chemical, so we'll try to keep it light. And it's really interesting. I actually put this talk together quite soon after with the COVID pandemic, and I'm really I say it's nice to share this technology and talk about some of the progress that we've made understanding the disease, but it's pretty crazy that we're still talking about this, over 2 years later. Yeah, so today I'll be presenting some of the more recent applications using these TMT reagents to study COVID-19, and specifically these reagents can help us identify, characterize, and quantify the protein levels during SARS-CoV-2 infection, as well as looking at some of the responses in different people, depending on how the disease takes hold. In addition, I'll talk about some of the more recent studies that use the TMT technology to understand the disease mechanisms. And I think that's the thing where we're still learning a lot. I mean, I think there's a lot of information about viral replication and which cells are infected, but really everybody's getting a different response. And even you can imagine it's changed with the different variants, as well as the different levels of vaccine status. So as things change, we're still using these reagents and studies, and there's even more data. I just saw a paper today that came out using similar technologies where they're trying to basically look at all the different combinations that are potentially out there. All right. So COVID-19 was initially reported as being a result of a novel coronavirus, SARS-CoV-2, in late 2019. By mid-March 2020, the COVID-19 was declared a global pandemic that's now spread to every country in the world. Since then, there's been over 530 million confirmed cases worldwide, 6.3 million deaths. In the United States alone, there have been nearly 84 million known COVID cases, almost over 1 million deaths. Unfortunately, these data only show confirmed cases. And also those are ones where you get tested by your public health agency, right? So with the advent of stay-at-home testing, as well as some of the people that have never been tested or were positive at the beginning of the pandemic when there weren't tests available, we know that these numbers are significantly higher, and due to differences in testing capabilities, but also just reporting criteria. So there's a lot of information we're still learning. Here, let me turn off my pointer here. I think it's kind of annoying. All right, 1 second. Let's see. Okay, we're back. here we go. So what's really puzzling is that with the individuals that are affected with COVID-19, they exhibit a wide variety of different symptoms and outcomes. Although a majority of people experience mild cold or flu-like symptoms such as fever, cough, congestion, fatigue, sore throat, nearly 20% experience more severe symptoms requiring hospitalization to manage. These severe COVID symptoms include things like blood clots, which can lead to stroke or heart attack, seizures, fainting, major lung, liver, kidney damage, and also hyperimmune disorders, things like encephalitis and some other autoimmune diseases. These later symptoms, which combined with underlying comorbidities, they are the ones that typically lead to patient death, right? So with healthy individuals, it's fairly rare that you'll have an infection that leads to a really bad outcome, but there are those that maybe have to be diabetic or have other underlying comorbidities that do affect that patient outcome. So really, this is the crux of it, is really what we're looking for are the differences. And so how do researchers find differences and to figure out which therapies you would want to use for different patients? And maybe we should use different therapies for if you have a mild treatment, right? Go home, rest up, drink lots of fluids, or more severe subjects, right? Where you basically have, if you can predict the outcome, maybe you need to be on an antibody therapy, or maybe you need to be taking a retroviral inhibitor. So these are the things that we need to understand. So it turns out that finding differences between individuals is really easy, especially ones that we can distinguish individual phenotypes, such as age, sex, weight, height. But finding disease markers is much more difficult because these differences must have strong correlation to the underlying disease state. But finding relevant disease markers is the hardest, right? Because you need to measure a lot of people, right? A lot of individuals using numerous replicates to get the statistical power to really determine if the thing that you're looking at is just something that's normally changing in human population or is really changing because of a disease or a particular therapy that you're trying to treat the disease with. And finding these statistically significant differences is actually the most challenging, and it requires, monitoring hundreds of thousands of samples. And that's where the clinical trials kind of start with a few individuals for safety purposes, go to phase 2, phase 3, and ultimately for phase 4, you're looking at whole populations of tens of thousands of individuals. So, the other thing I want to introduce here is that we want to understand the difference between accuracy and precision. And this becomes important when we're trying to determine differences, right? So just a reminder, accuracy describes a measurement error relative to the average. And in contrast, precision describes the measurement error relative to the standard deviation. And so high accuracy is really paramount to determining the true positives from false ones. But high precision is required to distinguish significant differences between conditions, right? So precision is really how we call a difference. Therefore, you want both, but even if you just have high precision with somewhat decent accuracy, it doesn't matter if the accuracy measurement is perfect, you can still call the difference if you have very good replicates and low CVs. So one method that we use in the mass spectrometry community is basically these mass tags. And so these are used to measure protein abundance with high precision, and they're called tandem mass tags. So these tandem mass tags, or TMT reagents, are used to label the samples and kind of barcode them so we can actually combine them together to get more sample throughput. Shown here on the right are the structures for the TMT 6-plex tags. And as you see, they're amine reactive tags, and they're identical in their overall shape and structure, right? So they have the same chemical structure and composition, but they do contain different stable isotopes of carbon-13 and nitrogen-15. And the isotope differences are also distributed, as noted by those asterisks, throughout the structure. So shown for the 126 tag, all of the stable isotopes are located in the linker region of the tag, whereas for the 131 tag, all the stable isotopes are located in the reporter ring. And I'll go through what that is in a minute. But basically these you can think about these as they all have the same structure and chemical composition, but they technically, if you break that reporter ring off, those reporter rings actually have unique masses, right? So the way I think about these is you can think of them as different colors. If you were doing fluorescent microscopy, and reading out, fluorophores, but in mass spectrometers, we read out the mass of the chemicals. And so therefore, they kind of look like these plate weights I've shown here on the left, where they're all the same size and shape, but they would have a different mass, okay? And in the past decade, my group and at Thermo, we've basically heavily invested and grown this technology. And we've expanded the original 6-plex set to 10, 11, and up to now 18 tags. And we've also been really focused on coming up with new reagent formats. These reagents are very hygroscopic and they're water sensitive, so they're difficult to use in humid environments. So we've done a lot of work trying to stabilize the tags as well as increase the number of tags so we can measure more samples at the same time. So shown here on the left is the latest set of reagents. We now have a set that we call the TMT Pro reagents, which supports multiplexing up to 18 different samples. In a typical workflow, the different TMT reagents are used to label different protein digests that then can be combined into one. And then once they're combined, we can then clean them up and load them into an LCMS instrument. Shown here is our Orbitrap Eclipse. And using this instrument, we can actually sequence the peptides. So shown here in the lower, kind of in the middle part of the screen is we have this MS selection where we select the peptides for sequencing. And during the sequencing of the peptides, we determine the amino acid sequence, but we also liberate those reporter rings. And those are the barcodes, and they actually allow us to tell us which sample that peptide came from. But not only that, we can actually look at the relative abundance of the reporter ion intensity and actually measure the amount of peptide that was actually from that sample. And so if you look on the lower right, you see this volcano plot, and this is basically where we do some statistical analysis to look at the upregulated and downregulated proteins for all the different proteins in the cell for all 18 samples at once. And the way I look at this is basically just like I described with my precision example, where we're just looking for things with relative differences in expression. So in order to measure protein abundances using TMT, and you're looking for these really small differences, you need to have good sample prep. And shown here on the left is the conventional mass spec method. And there's a lot more steps in mass spectrometry sample prep than there is, like, for instance, with cell staining or with traditional Western blotting, if you're familiar with that immunoassay technique. We have to lyse the cells, we have to sonicate them to disrupt the DNA and the RNA, and that's not very reproducible, and we have a lot of viscosity, because really all we care about is the proteins, right? And then those proteins, we need to reduce and alkylate them to break the disulfide bonds, because those cysteine peptides don't sequence if they're still linked together. And then finally, we need to clean up the protein to remove any of the detergents and the DNA and the lipids. We need to digest it to peptides for our, what we call bottom-up proteomics, so we can do the peptide sequencing. And then we need to clean up the peptides to remove any salts or anything that would carry forward through the sample prep. And so there's just a lot of chances that this method is pretty difficult. It's fairly time consuming. The traditional workflow takes 2 days and fairly tedious. But the big thing I want to call out is if you don't do your sample prep well, it really depends on how good your hands are. You can introduce a lot of variability. And you can imagine if you have differences in the sample input amount before you label with these mass tags, you will see a difference in the mass spectrometer, and it won't be due to the change in the sample. It'll be because you didn't pipette very well. And so some other sources of variability besides people are incomplete, sample lysis, reduction, alkylation, or digestion. You can have sample loss, poor reagent quality. I don't think many people think about that, but for mass spectrometry, we do need to use mass spec grade reagents, and also just the number of steps. And again, ultimately, these issues contribute to decreased reproducibility, and they can lead to low or poor quantitative measurements. So to simplify the mass spec workflow and improve reproducibility, my group has also been really focused on the sample prep, because the tags work really well, and we'll go through the workflow again, but we found that a lot of people just didn't make very good samples, and they were struggling to get good quantitative measurements. So one thing we did in the past 3 or 4 years is we kind of standardize and simplify the mass spec sample prep, and we call this the EasyPep mass spec sample prep kit. And in this new method, we actually combined a lot of steps together, right? So that reduced the number of, touch points, as well as the possibility of, the time it took to get through the sample prep. So the first thing we did is we actually, instead of sonication, we introduced a universal nuclease. And this enzyme actually degrades the RNA to DNA, so you don't have to, disrupt it using a sonicator. And then we also combined the reduction and alkylation step into a single pot 10-minute heating reaction. Since we actually control the lysis buffer going in, we don't have to do the protein level cleanup. And after digestion using a combination of trypsin and lyc, we can go directly to peptide level cleanup that not only cleans up and removes the salts, but it removes any excess TMT reagents, for instance. And so this new method, which typically compared to the old method, which took over 2 days, we can do sample prep for 20 reactions or 20 different samples in about 3 to 4 hours. So it's a big improvement. And unlike things like the genomic world where people are used to the mini prep kits, having standardized kits for the mass spectrometry proteomics is something I think we've seen a lot of other companies also get engaged with and have success with. So recently we also expanded the kits. So, we can do plasma, cells, tissues. We have the mini kit is our original spin column, but we also have a 96-well plate that we've actually have some applications with for automation, as well as a maxi kind of column, a large scale kit. And we use that kit for primarily looking at phospholipeptides, which are very low abundant and you need to process more sample for enrichment. And of course, we did check the performance of the different chemistries, and it's quite scalable. As shown here in this slide, you can see that between the mini, the maxi, and the 96, they all give very high numbers of protein identifications, shown here over 4,000. And we have very good digestion efficiency. And we measure this by looking at the miscleavages for lysine and arginine peptides when you cut with trypsin and lyc. And our spec was greater than 90%. And you can see that they all give excellent digestion performance. So back to the problem. So when you're combining this simplified, robust sample prep with TMT, it really does enable people to save a lot of time, especially when you're searching for biomarkers and when you have large sample cohorts. To illustrate this, in a typical label-free workflow, each sample or condition must be analyzed separately for all replicates. So in this example, when you have 18 samples and you're using 3 replicates each in a 3-hour LC-MS run, it would take 6 days, 24 hours, 6 days in a row to measure all of those samples. And you can do that what we call label-free quantitation. And it's a very accurate method, but as I mentioned, it's very time consuming. In contrast, we're using the TMT reagents. You can combine all those samples into one and run your 3 replicates in 9 hours. And this method enables the fractionation of the combined samples to give the same depth and coverage that you would get for the label-free analysis. So this is a huge time savings. And you can imagine during the early parts of the pandemic, this technology really allowed some of the researchers, and I'm going to highlight their work in a minute, to really get publications faster, to get data to the clinicians so that they can understand what was going on. So now I'm in the second half of my talk, I'm just going to highlight a few papers where people use this technology to actually identify differences. So this first one was looking at the viral infection, and this is the very first peer-reviewed paper on SARS-CoV-2 proteomics. And so what they use is they use TMT reagents to monitor SARS-CoV-2 infection in a model cell system. So these are the CACO2 cells. So their work involved harvesting proteins from the cells at different time points, and then they labeled those each time point with a different TMT tag. In this case, they used the TMT 11-plex reagents, and then they combined them in one sample, and they separated the peptides using this high pH reverse phase chromatography, and then LC-MS using this Q-Exactive HF instrument. And what they were trying to measure was the host cell proteins as well as the viral gene expression in the cells over time. So shown here is basically what they found is that their analysis identified and quantified an increase in the SARS-CoV-2 viral proteins over time, including the spike glycoprotein. And this is the protein that majority of the vaccine candidates were generated against, whether they were the protein-based vaccines as well as the mRNA-based ones. In addition, besides just seeing the viral protein expression increase, they saw numerous changes in the host cells, right? So it's not just, you want to obviously decrease replication, but you can imagine upon viral infection, the viruses hijack the endogenous cell machinery to make more viral particles, right? That's how it propagates. And in the process, it damages those tissues. And what they saw was that the host cells were actually having major changes in nucleic acid and cellular metabolism, which is not surprising because the virus is trying to make more virus and it usually hijacks those systems. In another study that was actually a preprint on the bioRxiv from Klein et al., they performed a similar experiment in, again, the same model cell system using the KCO2 cell models. But in this case, they were using the TMT tags to label replicate and mock virus cultured cells. And in this case, they wanted to look at cell signaling. So they did this phosphopeptide enrichment and before they did the high pH reverse phase. And in this experiment, they quantified over 7,000 proteins and over 15,000 phosphopeptide sites. So pretty good coverage of the cellular proteome. In this analysis, what really jumped out to them was changes in the RAS-MAP kinase pathway as well as AKT-mTOR. And that pathway is highly known for, or very well known for, protein expression. And so these protein pathways were activated upon SARS-CoV-2 infection. And so since they were upregulated, the researchers actually went back and found inhibitors against those particular proteins such as RAS, RAF, MEK, and PI3K. And they actually demonstrated in this paper when they added the inhibitors to those cells that were infected with SARS-CoV-2, it actually decreased the viral load. Now, these are not therapeutic compounds. They do shut down all cell signaling, but they were able to really show with the link with these inhibitors that you could control cell growth using these broad-based signaling pathway inhibitors. In another study, they looked at the host cell proteins that interacted with non-structural coronavirus proteins, specifically NSP2 and NSP4. In that very first publication I showed you, a lot of people were looking at the viral proteins that were basically for the spike protein or the cell coat. But there are a lot of other genes that are encoded by the coronavirus and we don't know their function. And you can imagine by knowing more about their function, we may understand why some people may have a different response to infection. It may not be the viral proteins themselves. It could be some of these other, exons that are present in the, or the other coding genes that are present in the virus. So again, since this is such a new virus, we don't know their function. So Davies et al. Performed a series of co-IP experiments. So they would overexpress these viral proteins because we don't know their function. They would put them in the cells and then they would pull them out to try to identify the protein interactions. And as controls, they actually use the common cold. So I thought that was quite interesting. So that's, CoV OC43. I never actually knew the common cold had a name, but that's the strain. And they also compared it to SARS-CoV-1. And this is that early virus, which is associated with MERS, M-E-R-S, which is a related coronavirus protein. And then, of course, they had control cells. So after tagging and expressing these proteins, they did the reduction, alkylation, digestion, and then the TMT labeling of those proteins. And what they were able to find was that there were some common interactors for NSP2, this non-structural protein, that was involved in endoplasmic reticulum calcium signaling. And it also affected mitochondrial biogenesis. They also identified interactors unique to each strain, such as a E3 ligase for SARS-CoV-1 and ER homeostasis factors for SARS-CoV-2. So again, related where these viruses are affecting different endogenous pathways. So the next study I want to cover are, again, more clinically relevant, where they're trying to understand why upon infection, people had different symptoms, right? So in this study, a lot of this work originally came out of China, and now it's been replicated in a lot of labs in Europe and the US, where they use TMT reagents to identify proteins that were upregulated or downregulated in different blood cell lineages. So in this particular paper, they were looking at PBMCs between healthy and mildly COVID symptomatic people. And they found that in mild infections, the majority of the proteins were unchanged. So if you see on this graph, anything that was statistically significant is kind of color coded by red or blue. And there's, a few hundred proteins that did change, but they weren't really jumping out at anybody. And again, this is due to mild infection. So you're not surprised there weren't some changes, but when they compared this to patients or subjects that were had more severe diseases, they found that over 200 proteins were significantly regulated, and majorities were involved with inflammation. So this has kind of been more recently termed this cytokine storm, where some people are basically, their immune system is working, but too well. And it's actually not only trying to fight the infection, but it's damaging the tissues, right? And that damage is actually caused by, can lead to some autoimmune diseases, but also can shut down other organs and cause, damages to your lungs. And so just showing here where CD4 and CD8, two very common markers for inflammation, were significantly upregulated for the more severe COVID subjects. Shown here was when they used tandem mass tags on the plasma. So again, not looking at the blood cells, but the plasma of healthy and control patients, or not patients, but subjects, using from asymptomatic to mild to severe. And in total, they monitored, they actually profiled 630 different plasma samples. And they were able to identify and quantify 469 plasma proteins, and with at least 3 replicates. And compared to the healthy control group, there were a few hundred dysregulated proteins. And specifically, they found that 120 proteins were upregulated and dysregulated in the non-symptomatic patient or people, but then 33 were downregulated in the severe COVID subjects. So again, this was kind of more of a broader study looking at numerous samples from a lot of individuals. So in that paper, they actually dug a little bit deeper, and although they didn't have a lot of individual samples that had overlap between individuals, because again, the disease I think is somewhat unique to the individuals, they were able to pull out specific pathways, specifically around coagulation, right? So the platelet pathway around aggregation, degranulation, and this blood clotting was actually something that also was commonly seen in people infected with SARS-CoV-2, where they were getting not necessarily damage from the immune system, but they were getting clots showing up in their lungs or causing strokes in some patients. So again, this pathway was also shown to be misregulated in SARS-CoV-2 subjects. And compared to healthy controls, they also identified SOD1 or a superoxide dismutase. And this was another marker. And so again, some of these proteins they were following up on to see if these could be biomarkers to predict which subjects would actually may progress to the more severe disease state, right? So you can imagine if it looks like this disease will become more chronic as we exit the pandemic phase, but you still don't know if you are actually going to have a severe case. And so if there could be a simple blood test, or we could follow up, and it's in addition just to looking at your viral load, we may be able to look at for biomarkers that could maybe predict or give your clinician or physician some indication of what type of treatment you should be receiving. So the last thing I would like to talk about is a really interesting application, and it's actually not just used for SARS-CoV-2, but this is actually one of the major applications of this technology, and it's actually for using it for drug screening. You can imagine when you want to look at the effect of a drug, you want to know its targets as well as any off targets that drug may be inhibiting. And this is typically done in vitro, where you have kinase panels and you can kind of look at the binding kinetics. But as you go into more, preclinical data sets, looking at cell culture models, and of course in animal models and people, you want to know, it's much more complex than those original drug screens when you started with, the initial primary screen. So this secondary screening technique is really trying to understand what are all the proteins that it's binding to. And so the way this works is we can do this thing called thermal profiling. So shown here on the left is you take your sample, and this could be a tissue lysate, it could be a cell lysate, and you treat those lysates with or without the drug, right? And then what you do is you slowly heat it up at discrete temperatures, and as you heat up the samples, the proteins that will start to precipitate, right? So they'll just like any other compound, they'll have a melting temperature and they'll denature and they'll precipitate. But you can imagine when you have something that's bound to the drug, it will actually shift the melting temperature, or the temperature at which it precipitates. And that's shown here on the right. So what we're looking at are melting curves. So In the past, we could only do this in like 8 or 10 samples, and we had to run them independently. But with these TMT Pro 18-plex samples, we can actually do this entire experiment in one sample. So we looked at all these different drugs, and this thermal profiling experiment allows you to analyze everything in about 2 or 3 hours, and there's fewer missing values, and it decreases your analysis time at about 50%. So this is a study that Freeman et al. Did where they actually, this had never been done before because this technology has only been used in the last 5 or 6 years. They actually went back and looked at approved antiviral inhibitors, right? So this was a very interesting area of research because we want to try to find, well, maybe there's an antiviral inhibitor that could maybe work for SARS-CoV-2, but nobody had ever done this thermal profiling approach to look at the off-target effects because there's side effects to every drug, but we don't really know all the targets so they decided to use this new technology to kind of screen them. So notably, what they also included a couple drugs that were popular in the news when COVID first became available. You have your hydrochloroquine, but also they looked at remdesivir, and this was the one that was developed and actually is the one that's only still clinically recommended for treating COVID. And then they also have ritonavir, which is now part of the combination for the Pfizer drug. But overall, they had 22 compounds tested, and they found that only about 34 proteins were significantly changed or stabilized by adding the drug. And you can see that upon each drug treatment, you get different profiles, and this is indicative of the different proteins that they target, as well as the off-target binding that could happen in the host cell. And one thing that jumped out in this particular study was a protein called TRP13. And this protein, is actually shown in red for remdesivir. And this was an unknown, but a newly discovered off-target. And this protein is involved in DNA replication and repair. And again, they didn't really link it to any side effects for this particular drug. But again, the thermal profiling was one way to basically show that this particular pathway could be impacted by taking this particular antiviral. And again, despite all this promising preclinical data, none of these protease inhibitors, except for remdesivir, have been approved for clinical use as a single drug therapy. There is now this Pfizer combination where they're basically using it now to treat COVID, but it seems like it kind of tamps down the viral load, but then it comes back. All right. So in summary, COVID-19 is a disease that's caused by this novel coronavirus, SARS-CoV-2. It's highly contagious. It results in a wide variety of symptoms with different levels of severity. One of the key tools that researchers have been using to understand the disease as well as the outcomes have been actually using proteomics in combination with TMT reagents, which allows you to quantify differences in protein levels with high precision in multiple samples at once that save both time and money. And overall, I hope my seminars highlighted the power of these reagents that provide clarity to the SARS-CoV-2 viral protein expression over time, changes in host cell signaling, immune responses in mild and severe cases, as well as the potential for COVID-19 drug candidate targets and off targets. And with that, I thank you guys for your attention, and I'll take any questions.
Thanks, Ryan. Very topical talk too. I can personally attest to this whole rebound thing on Paxlovid. That's no joke.
Oh, really? Okay, yeah. It's out there, and
yeah,
I think they definitely could extend it and possibly, again, they're rushing things, but I think it's working. So that's good that there's some therapies out there.
That's true. Yeah, for sure. So any questions for Ryan? You can same trick. I think you could probably just unmute. We have a great turnout, but it's a small enough group. Probably you can chime into the question or post in chat. Ryan, can you see the chat?
Yeah, bringing it up.
All right,
here.
You've got one question in there.
Oh, that's a long question. All right. It says, given the high prevalence of severe cases, is there a high prevalence of the diseases there's now in reinfections? How can this protocol efficiently and feasibly be applied at a national scale? Is there a components analysis for COVID that identifies the prevalence of severe infection prior to? Yeah. That's a really good question. I think, unfortunately, this is a research tool. It's not something that's clinical mass spectrometry is, I think, if you guys want to have me come back sometime, we can talk about that. A lot of this is still in the lab, right? There are some point of care and clinical mass spec analyzers, but they're primarily used for typing microbes or looking at small molecule like concentrations in your blood, like testosterone or vitamin D. They don't really use these technologies in the clinic, but these are definitely used by some consortiums, specifically around cancer research. Earlier, we did get into some, there was some ideas about using proteomics to help, like at the national scale, like the UK government was like, we want to screen every single, COVID sample, right? And they were doing all the genomics and getting the idea of which strain, but you can imagine that's only part of the picture. And I think getting information about the person, right, getting some personalized medicine would be ideal. Unfortunately, I just don't think we have the infrastructure to support it yet. And the technology is still quite expensive and requires a lot of experts to use. And that's a big focus of my group, is we call it standardization is the first step, and we call the second one democratization, right? If we can lower the barrier to entry, I think it would make this technology much more accessible to biologists, for instance. Mm-hmm. I don't know if to answer your question, but it's a big question. Yeah.
You got a thank you in the chat, and then another quick question, a shorter question.
Yeah, cost reduction. That's an interesting question. So we get that a lot. So the tags are not cheap, I'll be honest. The kits are quite expensive. So just to give an estimate, they're about $8,000 for the set of the 18 tags. That can actually label, though, about 1,000 samples. So and that's the bulk size. So you're looking at about $8 a sample. So for your, that's the additional cost of using the tags versus doing no tag, right? So labeled versus unlabeled samples. So there's a cost to the technology. But that being said, if anybody's ever submitted samples to a core lab for proteomics, they charge about $1,000 an hour to run the instrument, because the instrument is, at least for TMT technology, it requires an Orbitrap or a higher resolution mass spectrometer. They're $500,000 to $1 million instruments, much like, some of these other expensive platforms. And just like, cryo-EM, not bad. That's not as bad as cryo-EM, but it's up there in terms of cost. And so if you're spending $1,000 an hour, if you wanted to do 6 days, 24 hours, right, that study is out of reach for most researchers, but they can buy a kit and get the data in 9 hours. So you're looking at, $9,000 kit or $8,000 kit, $9,000 worth of mass spec time. You get the same data that you would normally have to spend at the core lab for a whole week's of analysis. So core labs like this is one way to do it. There's obviously other ways to measure protein abundances. If you know what you're looking for, you can always buy isotope labeled peptides that you can spike in. So that's another thing that my group supports. It has its place and it's very powerful, but for some applications, it's definitely overkill. Yes. Ask a question about adapting it for glycopeptide analysis. So glycopeptides work well, and actually there's a lot of publications using glycopeptide analysis with TMT reagents. And one cool thing that's kind of a trick that's known in the community is the glycopeptides are very hydrophilic, right? And so they're difficult for the mass analyzer to, especially if you're doing LCMS, when you do the chromatography part with C18, the glycopeptides, they don't retain on the column very well. And also in the mass spectrometer, they don't ionize well because they got the sugar. And one trick is when you put these mass tags on the peptides, the glycopeptides in particular, it makes they're more hydrophobic because the tag's on there, and they also ionize better because there's some amines in the peptides, and so they ionize better. So actually there's a lot of glycopeptide workflows that use this TMT reagents. And the funny thing is sometimes they don't even use the multiplexing. They just stick the tag on just so they can get better MS data. Mm-hmm. Yep.
Thanks, Ryan. I do not see any other questions in the chat or hands raised.
All right.
Okay, and then so thanks again for an awesome talk. It was great turnout from everybody today too. Before we wrap up, Tony, do you have any announcements? Anything we want to say about upcoming sessions?
Well, we are looking right now. It looks like we are going to be postponing the July session because of it was originally scheduled at the right next to the July 4th holiday, and also because the University of Oregon's campus is going to be really difficult to get into during the World Championship, so we couldn't really push it later in the month. So our next scheduled workshop is going to be for the first Wednesday of August. And yeah, we'll still have more information coming on where that will be, and hopefully we'll be able to do that in person again.
Sounds great. Thanks, Tony.
Great.
I hope everybody has a good end of term on our end and a great start to summer for everyone. And see you all, it sounds like in early August.
It's good. Adios.
All right.
Have a
Bye,
great
everybody.
day.